Hi, my name is Lucia and myself and my colleague Roma Crooks will be talking to you today about ACE inhibitors, enzyme inhibition, the renin-angiogenesis cycle, and recent studies which have shown to support the theory that luxuries such as chocolate, wine and tea could actually help in the reduction of blood pressure and therefore lower the requirements of medical intervention.
So, we all know about enzymes, but just to recap, enzymes are proteins and biological catalysts. They increase the reaction rate, converting a substrate into a product. Inhibitors, which come in a variety of different forms, decrease enzymatic activity. This has been manipulated by the pharmaceutical industry to produce drugs to treat a variety of human dysfunction, ranging from depression to hypertension.
Enzyme inhibitors are molecules which decrease the enzyme's activity by blocking the active site on the enzyme and therefore preventing the substrate from bonding to produce the ES complex. These inhibitors can cause inhibitions which are either competitive, uncompetitive or non-competitive. These terms are simply to describe the interactions between the molecules. Many drug molecules are in fact enzyme inhibitors, depending on its potency and specificity.
The purpose of ACE inhibitors is to reduce blood pressure by inhibiting the conversion of angiotensin 1 to angiotensin 2. The renin-angiotensin cycle maintains blood volume and blood pressure. If these were to decrease, a chain reaction is set into place. A reduction in renal perfusion is detected both locally and systemically. The juxtaglomerular apparatus in the kidneys causes the release of the enzyme renin. Angiotensinogen, which is in constant production by the liver, is then converted by renin to angiotensin 1. As angiotensin 1 passes through the lungs, it is acted upon by angiotensin-converting enzymes, also known as ACE, and converted to the vasoconstrictor angiotensin 2. ACE also inhibits the vasodilator bradykinin in a dual attempt to increase blood pressure. Therefore, inhibition of ACE depletes the inhibition of bradykinin, the vasodilator, and prevents the conversion to angiotensin 2, a vasoconstrictor. This prevents a blood pressure raise.
ACE inhibitors are originally synthesized from compounds found in the venom of the pit viper. This species of snake detects its prey by the sense of body heat and determine the size of its prey by its body temperature. How ironic is it that something which was intended to kill something else has resulted in the saviour of millions? The ACE inhibitor inhibits ACE, which is the angiotensin conversion enzyme, a major component of the blood pressure regulating the renin-angiogenesis system. Frequently prescribed ACE inhibitors include captopril, which is often called capotin, enalapril, lisinopril, a drug which can also be used in the prevention of renal and retinal complications of diabetes, and lastly, ramipril. ACE inhibitors, abbreviated from angiotensin conversion inhibitors, lower blood pressure and are frequently used in patients which have suffered hypertension, which is sustained high blood pressure, and congestive heart failure, which is the inability for the heart to provide inadequate blood supply.
The majority of ACE-inhibiting drugs bind to the angiotensin-converting enzyme due to interactions between the zinc in the enzyme and the bonds in the inhibitor. It was found in clinical tests that the C domain of ACE was mainly responsible for the regulation of blood pressure as opposed to the N domain. This suggests that inhibitors selective for the C domain would be most useful at preventing the cleaving of angiotensin 1 to angiotensin 2. However, bradykinin is hydrolyzed by both the C and N domain active sites, so a C domain selective inhibitor may prevent the build-up of excess bradykinin, which is a cause of angioedema, a side effect of ACE inhibitory treatment. ACE activity was assayed in the presence of chocolate extract in the rat kidney membrane. It was noted that the inhibition was dependent on the content of flavanols present. This suggested it was flavanols that possessed the ACE inhibitory qualities. Other foodstuffs containing flavanols include wine and tea.
A recent scientific article in a journal had been titled Effects of Cocoa Extract and Dark Chocolate on ACE and Nitric Oxide in the Human Endothelial Cells and Healthy Volunteers. The scientists behind the experimentation were trying to prove that the evidence which had previously been suggested that cocoa from the bean Thromboma cacao L is actually beneficial for the effects of cardiovascular disease. They aimed to investigate that if cocoa extract and dark chocolate did in fact influence ACE and nitric oxide in vitro in the human endothelial cells and in vivo in healthy volunteers. The ACE activity was analysed with a commercial radioenzymatic assay and measured in human endothelial cells from umbilical veins after 10 minutes of incubation with cocoa extract.
So in conclusion, enjoy wine, tea and chocolate, all in moderation of course, and thank the ACE inhibiting qualities of the flavanols in these products. Thank you for watching.